Hi, I'm Jay Phelan. Let's learn some biology. We've been talking about the nervous system, and I want to talk now about a surprising and really interesting feature of the human brain. Way back in 1954, a psychologist, James Olds, was working with some rodents, rats, and he took electrodes and he was putting them into different parts of the brain and giving them a little bit of stimulation and evaluating what happened. Usually nothing happened, or he couldn't tell what happened. But he then discovered that there was a place that if he had the electrode there and he stimulated it, he felt that the rats liked it. He was anthropomorphizing, but he said it was pretty clear they liked it. He tested this by constructing different things that they would have to do in order to get rewarded with stimulation of this part of the brain. So for instance, if he put them into a complex maze and they only got stimulation in that part of the brain, it's in the mesolimbic area in the center of the brain. If they got stimulation from there at the end of learning this maze, they were motivated to learn it, and they could learn to navigate extremely complex mazes. Here we have one of the pioneers of this early research, Claude Shannon, showing one of the mazes that they used. James Olds also discovered that if he constructed a setup where the rats could push a lever and they could give themselves stimulation in that part of the brain, in the mesolimbic area where he had implanted the electrodes, that they'd push the lever, push the lever, push the lever, push the lever. This behavior was telling them they liked it. And in fact, they would push the lever and push the lever repeatedly without stopping. He had some rodents that were in cages where they had another lever right next to it, and if they pushed that lever, they would get a pellet of food. When they had these two choices, what did they do? They pushed the lever for stimulation of that part of the brain that he knew gave them a lot of pleasure, and they'd push it and they'd push it and they'd push it without stopping, as much as 7,500 times in 12 hours. If he gave them free access to push it as long as they wanted, they would keep pushing it until they died from starvation. So they couldn't even take a break for long enough to push the other lever, get a little bit of food, and then go back to the pleasure lever. That's how motivating and rewarding it was. He thought of this as a pleasure center, but sometimes we call it a do-it-again center, because an organism, if it has the ability to get stimulation there, will do whatever it has to do in order to get stimulation there again. That causes you to think of the question then, why are we built this way? What good would it do an animal to have neurons in a region in its brain cause feelings of pleasure when they are stimulated? What good would that do? Why would we be built this way, just to have this sensation of pleasure? Now think about it. So your brain is constructed from cells, and the instructions for these cells and how to make them come from genes, and the genes are the products of natural selection. Where's the adaptive value of having these do-it-again centers? It provides a way that you can motivate the animal. So genes, these sequences of DNA carrying instructions for how to build the animal, they can create a reward system to incentivize the animal. What are the types of behaviors that we would expect then through evolutionary time to become associated with stimulation of the pleasure centers? Are we going to get lots and lots of stimulation of these reward centers in our brain for solving math problems? Probably not. For doing puzzles? Probably not. What about finding food? What about having sex, reproductive success? What about achieving status, something that is going to increase an organism's access to mates? All those things, tremendous fitness value, and in fact, all of those are associated with lots of stimulation of that part of the brain, the do-it-again centers in the brain. What's going on there? We've got several of these throughout the brain, 3 or 4 of them. Typically what we see is this. So we look at our showerhead model of terminal buttons at the end of axons in these parts of the brain. in the pleasure centers, the do-it-again centers, the neurotransmitters that are filling the vesicles are filled with dopamine. So what happens is this. The neuron is stimulated in response to your behavior, finding food, finding status, winning in some competitive interaction, and so on. The neuron gets stimulated from that. Dopamine gets released into the synapse. It floats around. It binds to receptors on the postsynaptic cell, causing that cell to fire. Happiness ensues. You feel good. You want to do again that thing that got you that release of dopamine. That's how it works. So we can link that to lots and lots of different sorts of behavior. It can be a sort of an all-purpose reward center. Now here's a very unfortunate coincidence. In these parts of the brain, in these do-it-again centers, where dopamine's going to be released. And you can sort of think of it as, at any given time, it's like little nozzles that are turned on, rather than just, oh, you did the behavior, here's the release of dopamine. It's like little nozzles that there's always some going on. Because think about it, right now, wherever you are, you have shelter, you have clothing, you've had some food. You're doing a lot right in your life. You're taking a class that's going to make you smart and learn how to be better at life. So you got dopamine going on. That's always happening, and the nozzle gets turned up or turned down in response to these behaviors. But just through an unfortunate coincidence, cocaine, the drug cocaine, happens to be very similar in shape to dopamine. And when dopamine gets into the synaptic cleft, the space between these neurons, it can bind to the reuptake receptor. So remember how typical neurons work. They release the neurotransmitter, binds to the postsynaptic cell, triggers something, gets released, and then we get enzymatic degradation and reuptake. So cocaine binds to the reuptake receptors, and what that does, by sitting in the reuptake center, it doesn't get taken back into the cell, it just sits there. So now dopamine that's being released floats around, triggers the postsynaptic cell, it fires, now it diffuses back. Instead of reuptake occurring, it just keeps floating around, and it hits the postsynaptic cell again. Bam, it fires again, and it gets released, floats around, reuptake doesn't occur, floats back, hits the postsynaptic cell receptor, bam, fires again. So by blocking the reuptake receptor, you get an intensification of the pleasure that normally would be occurring because of this dopamine. So we can look at, through these MRI helmets, where we see how much activity is in different parts of the brain, and we can look. Here's one measure of what's going on in a brain in a person who has just done cocaine. And you see right in the center of the brain, in the limbic system, where these cells are that James Olds initially found, tremendous activity. The intensity of the coloration is how much activity. So in the reward center, huge amount of activity going on. Elsewhere in the brain, not so much. This is the response of your brain to cocaine. So essentially, there's a message that normally, because of dopamine release, your brain would be thinking, hey, we're doing the right thing, we're getting status, we're finding food, we're getting mates, and so on. So you're getting that message. It doesn't get terminated. You think, I got to keep doing this. But in this case, it's because there's cocaine in the synapse. So it's gotten hijacked, essentially. This then shouldn't surprise you that there's a big link between dopamine parts of the brain, dopamine receptors, and addiction, right? If there are going to be things that cause more activity there, then people are going to want to do it again. So we see a huge amount of research here as we try to understand, how can we modulate this? How can we try to help people? Because when you do drugs like this, it's hard, you can't just say no, because your brain is getting the message that says, no, this is actually the right thing to do from an evolutionary perspective. So there's lots of research looking at which receptors cause people to maybe be more susceptible to the benefits of, or the pleasures associated with, drugs and other things that go along with addiction. It's still a rich area of research, but we know that there are lots of tight links between dopamine and drug addiction. So the important take-home message is that our brains are built with pleasure centers. From an evolutionary perspective, this is extremely important. Stimulating these pleasure centers is so pleasurable that we want to behave in whatever ways repeat and maximize that stimulation. Sometimes this ends up with bad outcomes. In our evolutionary past, though, this was a really good way to build in a motivation system for influencing the behavior of organisms.